On May 15, 2022, the New York Times published an article titled "Cannibalistic Toads Reveal Evolution in Fast Motion, Study Finds" by Annie Roth. The New York Times article essentially states this: cane toads, a large, poisonous, warty species of toad, are native only to South and Central America. However, in 1935, cane toads were introduced in parts of Australia in an attempt to decrease the number of cane beetles, which were becoming a problem for cane farmers at the time. Because the toads had little to no predators in this environment, their populations rose quickly. They have become an invasive species in Australia and have squeezed out many native amphibian populations. Over time, researchers noticed something very peculiar about the behavior of these cane toads. The tadpoles were commonly found consuming the eggs of their own species, engaging in cannibalism. These cannibalistic tendencies had never been found among cane toads in their native lands, so why was this suddenly happening in Australia? How did this cannibalistic behavior evolve so rapidly? For decades, these questions puzzled scientists, until recently when a study was published that may have discovered the answer. According to the New York Times, the study found that the reason the tadpoles were attracted to the toad eggs could be because of a chemical present called bufadienolide toxins. This is the same chemical that gives the toads their toxicity, protecting them from predators. They say that the study exposed cane toad tadpoles to the toxins and saw that the tadpoles would consume eggs of all amphibian species when they were exposed to the chemical. They claim that the study attributed this rapid evolution to being a mechanism for reducing interspecific competition against other cane toads within their environment. Now, we move on to the summary of the primary research article referenced by the New York Times article. The research article by Michael Crossland, Richard Shine, and Jana DeVore is titled "Choosy Cannibals: Targeted Consumption of Conspecific Hatchlings by Larval Cane Toads is Triggered by Species-Specific Defensive Toxins." This article gives a more in-depth scientific description of the experiments conducted to discover if bufadienolide toxins are what caused cane toad tadpoles to engage in cannibalistic activities. In order to ensure accuracy, the researchers conducted a total of 3 experiments. The first experiment tested whether or not cane toad tadpoles would consume the eggs of hatchlings of other amphibians that were native to Australia. They placed one tadpole in a container with 10 eggs of another species of amphibian and recorded the survival rate of the amphibian hatchlings. As you can see in the graph, the results were just as they expected. There were very small proportions of predation on the native amphibian eggs compared to the cane toad eggs. The second experiment aimed to test whether or not bufadienolide would increase the cannibalism of toads on eggs of the same species. Both treatment groups included 10 cane toad eggs, 1 tadpole, and a ceramic ring. One group had no toxins, and the other group had a ceramic ring that emitted 2 milligrams of the toxins. The results are shown on the graph, with the black dots being the proportion cannibalized with the toxins present and the white dots without toxins present. The proportion consumed was 3.31 times higher when the toxins were present. The third experiment tested whether or not the toxins would increase predation on eggs of other species. Each treatment group had 1 tadpole and 10 eggs of this frog species known as Tourneurs's frog. The first group was a control. The second group had 1 milligram of fresh bufadienolide mixture, and the third group had 1 ceramic ring emitting 2 milligrams of the toxin. The results are shown on the graph. The researchers concluded that the predation rates on unhatched eggs were low in the absence of the toxin and high in the presence of the toxin, regardless of the prey species. Overall, the researchers concluded that the tadpoles were highly attracted to the toxins, which they believe displays a mechanism for cannibalism that evolved as a result of extreme population growth of the cane toads in Australia. The researchers also stated that they believed their results were widely applicable for cane toads across all of Australia. Now, we'll compare the primary research article and the news article to see how accurately the study was conveyed in popular media. The biggest difference between the research and news articles is that the research article was more in-depth. Obviously, it would be difficult to convey every detail of the study, but there were some key aspects that were not addressed at all, like the 3 separate experiments that were conducted. Instead, the news article summarized the 3 experiments to sound like it was one cohesive experiment, and did so in a way that wasn't entirely accurate. The news article made it sound like both species of egg were in the same tank, when the reality is that these 2 were completely separate experiments done with different tadpoles. The news story also focused a lot more on the history of cane toads in Australia and other information obtained from outside resources. There were many quotes from the authors of the research study that weren't mentioned in the research article, but none of them were inconsistent with the data published. Overall, the New York Times article was a fairly accurate representation of the findings of the study. Although every detail wasn't there, the main message that the article wanted to convey was intact and the information was accurate for the most part. The reason some parts weren't as in-depth is likely because the length of the news article was limited, and the writer wanted to keep the reader's attention. Despite its lack of detail, the news story was mostly accurate to the study and represented a good way to diffuse scientific findings to the public and make complex information more accessible.